All right. So welcome, everyone. Thank you so much for joining us today. Before I start, I would like to acknowledge the Gadigal people of the Eora Nation, who are the traditional custodians of this land where we are broadcasting the seminar from today, and pay our respects to the elders, both past and present. So today we're very happy to welcome a Mexican speaker, Dr. Santiago Ramírez Barahona, who's speaking to us directly from Mexico City tonight. So Santiago is actually a specialist of tree ferns. He's done his PhD at the University of Mexico in UNAM on tree ferns, on the ecology of tree ferns. He's then gone on to a few postdocs in the US, in Veracruz, and most recently at UNAM with Susana Magallanes, working on the paper that he's going to present today. So now Santiago is an assistant professor there at UNAM, and we're very grateful to have you today. So off to you.
Thank you. Thanks, Hervé, for the invitation for this virtual seminar. Can everyone hear me all right? If not, just say so we can figure things out. And so yeah, thank you, Hervé, and thank all of you for joining in. For this virtual talk, I'm going to give about the delayed rise of flowering plants. It's basically the result of my work as a postdoc with Susana Magallanes and in collaboration with Hervé. And this talk about flowering plants, well, to start off, we all know that, oh, come on, something is, oh, there it is. We all know that angiosperms are by far the most diverse group of terrestrial plants, by far the largest group, with over 300,000 described species to date. But this hasn't always been so. There was once a time where there were no flowers. And angiosperms, flowering plants, have a really interesting evolutionary history. The first evidence, if we look at the paleobotanical record, the first evidence of angiosperms comes from the early Cretaceous. At around 125 to 140 million years ago, there's the evidence of the first angiosperms appearing in the form of pollen grains and also remains of other macrofossils. The curious thing about angiosperms evolution is that once they appear, suddenly they start to diversify dramatically in terms of taxonomic diversity, in terms of morphology. Here you see a scheme from Herendeen and colleagues from 2017, where you see the different kind of schematic of the different floral rearrangements that we see in the fossil record of angiosperms, and it's fairly evident that by really quickly before they appear, angiosperms really rose in diversity. One curious thing is that angiosperms are the last group of extant plants to appear, but they rapidly became very diverse. And when you look at the fossil record, their taxonomic diversification is basically clustered or occurred during the Middle Cretaceous, very quickly after they appeared in a relatively short period of time. If you go to the fossil record, more than 50% of extant families that we can recognize in the fossil record appear prior to 70 million years ago. And if you look at molecular phylogenies and molecular age estimates, they give the same picture. Over 75% of extant angiosperms originated prior to 70 million years ago. So this patterns or these trends that we're seeing in the fossil record are extraordinary, and they appear to hold telling us a really interesting picture of angiosperm evolution. This dramatic rise in phylogenetic diversity is also mirrored by the geographical extent angiosperms. Here you see a plot in the y-axis is millions of years ago, starting in around 140 million years ago. And in the y-axis, you see latitude. And then you will see how angiosperms first appear at lower latitudes around the equator, around the equator, and then very rapidly starting to colonize basically throughout the world, from Victoria and Antarctica, Queensland, all the way to the Canadian Arctic. So basically, angiosperms appear subtly, diversify quickly in terms of species numbers, in terms of morphology, and also geographically, angiosperms start to be kind of a conspicuous element of terrestrial ecosystems really quickly. However, there is a great deal of paleobotanical evidence that suggests that, yeah, by the late Cretaceous, angiosperms were very diverse and widespread, but they were still not ecologically dominant. Here you see one of the classic plots of angiosperm species, relative species abundance. Sorry for that. Species abundance in terms of relative abundance in the y-axis, and in the x-axis you see the age from 145 million years ago to 65. And the interesting thing is that they rise in diversity, but still by the end of the Cretaceous, they were not the dominant group of plants. Around 40% of all species belong to angiosperms. So there were a lot of ferns and gymnosperms and other groups. So there remains questions about the timing and obviously the geographical setting and the groups that were involved in the rise to ecological dominance of modern-day angiosperms. And more specifically, what we aimed with this work, our main question was to see whether the initial appearance of most families during the Cretaceous was followed shortly by their diversification into extant species. So this is basically what I'm gonna address here today. But before that, it is important to recognize the distinction between stem and crown nodes and what these are telling us. So in essence, every group of organisms, in this case, angiosperm families, have two very important nodes, two very important ages. The stem age, which marks the divergence from the sister group, and the crown age, that you can visualize this as the marking the most recent common ancestor of all living species. So with the proper sampling, we can get here depicted by this white circles. With the proper sampling, you can trace back the stem node of families, the age of origin, the age of divergence of these families. But if you add a couple of other selected species into the sampling, you can also trace down the crown node of each family. And this is very important because, as I told you, by tracing these two important nodes in the phylogeny of a group, we can trace the divergence with the sister group, but we can also trace the most recent common ancestor of its living species. And also, we can compare these two and estimate what has been called the phylogenetic fuse, which is nothing more than the time elapsed between stem and the crown age of a particular group. So, given this context, what we wanted is to estimate estimate both stem and crown ages for angiosperm families. So where we stood when this project began was we wanted to understand the temporal patterns of angiosperm diversity, and we relied upon the phylogeny of Magallanes and colleagues from 2015. By then, it was the largest family-level phylogeny for angiosperms. However, not all families were included in this tree, although it was a really good sampling. Around 85 to 87% of families were represented in this tree by Magallanes. But most of these were represented by single terminals, including the mega diverse families such as the grasses and the orchids and the legumes. And this is important because we couldn't trace the crown age of these families. If you look at the representation of crown nodes in this tree, a little bit over 24% of families were represented by the crown nodes. So what we wanted to do was to increase the taxonomic sampling and try to bring these numbers up. So we went to the Angiosperm phylogeny website. Many of you might be familiar with this website. It's really amazing work. And we revised thousands of family level phylogenies for angiosperms. And the first thing is that we brought our taxonomic sampling all the way up to 100% to include all currently recognized families, and also to have most of the largest here in the yellow box. All the major, the largest families were represented also by subfamilies in our taxonomic sampling. And most importantly, in terms of the species, the families that we have with crown nodes, we're able to increase this to around 85% of families with crown nodes represented by our new taxonomic sampling. And also you can see in yellow, we had the crown node for at the subfamily level for the largest families. That's basically families with over 2,000 species each. So long story short, we managed to get all families sampled with crown nodes for most of this. And we produce this tree. This is a maximum likelihood tree. Here are some numbers for the tree. 1,209 tips, chloroplastid and nucleogenes. And the next step that we wanted, and this is really what we wanted to get, is to use this tree. We had all the families, we had a good resolution, good data. We wanted to date this phylogeny. And for that, we went to fossil calibrations. You know, fossils are used to time calibrate phylogenies in terms of absolute time. But most studies dating angiosperms were used less than 60 fossils, with the exception of the phylogeny from Magallanes and colleagues from 2015 that used 136. So basically, we wanted to expand on this fossil sampling of Magallanes. And what we did, we went through a lot of paleobotanical literature. We went to the original references describing the fossils, and we basically captured all the information, documented every fossil entry in our database, and ended up with 238 fossil calibrations that were justified following best practices. If you like, our paper has a really long PDF showing all the justifications for each fossil. So we use this 238 fossils, which is a great improvement over using 52 or 24 fossils for calibration. And what we did, we combined that with our phylogeny, and we produced this dated phylogeny for all angiosperm families. I'm going to go into detail of the phylogeny, but this dated tree, but there are all the major groups of angiosperms, all the families with their crown nodes. So what we did was to then extract, for every family, we extracted the stem age and also the crown age. And what we found for stem ages, you see here the probability density of stem ages through time. And what you see is something that was kind of not surprising, what we're seeing with other phylogenies, what we were seeing with other with paleobotanical data is that around 59 to 80%, this is depending on calibration strategy, around 59 to 80% of families have their stem age in the Cretaceous, especially towards the late Cretaceous. And again, this kind of coincides with what we know from the fossil record. But interestingly, when we looked at crown ages that we basically didn't know when these crown ages were distributed through time, we get, again, depending on calibration strategy, around 60 to 90% of families' crown ages are within the Cenozoic. So a very basic insight is that this tells us that most families diverged in the late Cretaceous, but intrafamiliar diversification is a Cenozoic phenomenon. And the striking thing is that when we compare these two, when we put this together and kind of go back to this idea of the phylogenetic fuse, and we wanted to test how different are stem ages and crown ages for every single family. So we estimated phylogenetic fuses, and we obtained these results. Here you see in the y-axis the number of families, and in the x-axis the length of the phylogenetic fuse in terms of millions of years. And we found, again, depending on calibration strategy, that the mean phylogenetic fuse of families was somewhere between 37 and 56 million years. That is, once they originated, Angiosperm families took around 40 to 50 million years to start their crown diversification. So the next step that we wanted to do is, okay, we know how these phylogenetic fuses are strikingly long. You can see some families that took a while to have their crown diversification. And the most straightforward question was to see how are these phylogenetic fuses distributed throughout the Angiosperm phylogeny. So here you see the same phylogeny with the branches colored by categories of fuse length. That's short fuses, intermediate fuses, and long fuses. And what you can see in this evidence is that short and long fuses are basically randomly scattered throughout the phylogeny. There's no evidence of clustering of the length of the phylogenetic fuses. So what this is telling us, or this is our interpretation, is that this pattern of phylogenetic fuse appears to be widespread throughout angiosperms. It's not just a couple of groups that are pulling this estimates. So our question was, why are angiosperms on a broad scale appear to be have been delayed in their diversification? So one avenue that we took to answer these questions was to look at the spatial patterns of Angiosperm family diversity. And we took publicly available data from the Global Biodiversity Information Facility, around 60 million records associated with urban specimens throughout the world. But there's a lot of problems we encountered. The GBIF data is not cleaned. There's a lot of synonyms, there's a lot of bad coordinates, identification problems, and also the problem with non-native occurrence of species. So if you take GBIF data and plot it, you get this kind of noisy picture, and you can notice there's a lot of noise going on. So this is not good. So what we did was to kind of impose several taxonomic and geographical filters to this data using several online resources. And we get this picture, sorry for the bouncing between slides. So we get this picture. We acknowledge that it's not perfect, but it's much better. It gives us quite some decent geographical distribution across the world. And so we took this data, and the first thing that we did was to do estimate species richness and family richness across the globe. And here I'm showing you a map of family richness across the globe. And darker colors indicate a higher number of families within particular grid cells. And the first thing, it's evident that the pattern, the mega diverse tropics of South America, Central America, Africa, for example, Madagascar, Southeast Asia, New Guinea, are places with a lot of families. And this correlates nicely with diversity at the species level. So we wanted to take this spatial information and integrate it with our data phylogeny, with our temporal patterns of family ages. And for this, we estimated this index, the mean family age, on a per grid cell basis. And basically, what this index tells us is, on average, how old are families occurring within specific grid cells. So for every grid cell, we had a mean family age. So we did that. The first thing we did was to estimate that for stem ages, and here you see the results in the map. You see darker colors in red correspond to older mean stem ages of Angiosperm families, and it's evident that tropical biomes or tropical regions have older family stem ages than other non-tropical areas. When you take these estimates and group them by these three biome categories that we constructed, arid, temperate, and tropical, it is evident that tropical biomes have on average older Angiosperm families. But this is not quite so clear when you look at crown ages. Again, you see the same map in blue. Darker colors indicate older crown ages for families. And you can see there's places like New Guinea, Southeast Asia, tropical Australia, Africa, parts of Central America that are showing up as having older crown ages, but the pattern is less clear. Again, when you look at biome, these three great biome categories, there's really not a pronounced difference between crown ages across these different biomes. So you may ask, and this is what we ask ourselves, what about family phylogenetic fuses? How are these distributed throughout the world? So we basically combine these two and we get this picture. In green, again, darker colors indicate longer phylogenetic fuses. That means, for example, in the Amazon, there are families that have an average longer phylogenetic fuses than, for instance, in Patagonia or in the southwestern North America. And when you look again at these three biome categories, it is clear that tropical biomes have families, or tend to have families with longer phylogenetic fuses. And this kind of makes sense if we go back to what we know, what we know of Angiosperm families from, of Angiosperms in the paleobotanical record. We kind of think, or evidence suggests, that the first Angiosperms were tropical, and this is again suggested. Here I show you a paleogeographical map of the early Cretaceous. Perhaps the details are lost. They turned out to be too small, the map, but believe me, just focus on the four black arrows that are depicting the approximate location of the first fossil Angiosperms, and they are located within tropical and border tropical regions that have been inferred to have been distributed back then in the early Cretaceous. So this kind of makes sense. Angiosperms originated in the tropics, and that's why we're seeing this old ages and longer phylogenetic fuses in these areas. So our next question was, we wanted to actually test this hypothesis and look at how, if we could test this hypothesis by doing ancestral state reconstruction using information on the sample phylogeny and geographic distribution across these biomes. So we did an ancestral state reconstruction, which I'm showing you here, the same phylogeny. The color of the branches indicates the inferred ancestral state, either temperate in blue, tropical in yellow, or arid in red. And here the insert pie charts indicate the most likely ancestral state for keynotes along the backbone of the Angiosperm phylogeny. And as you see, and this again is what we suspected, is that the most likely ancestral biome of angiosperms is indeed tropical. But looking at more detail into this phylogeny, there's some interesting patterns. For instance, the earliest transitions that we can date there into non-tropical biomes, for example, in Caryophyllales, this is one of the oldest transitions into arid environments, or again in Brassicales. These transitions occurred 40 to 60 million years after crown angiosperms. So again, this kind of makes sense to what we're looking in the map, that angiosperms originated in tropical environments and really took a while to be able to colonize and to go out of the tropics and diversify into this non-tropical, arid and temperate environments. Moreover, this tells us that the diversification and ecological expansion of extant angiosperms was geographically heterogeneous and perhaps was fairly quicker in non-tropical environments. This is, I failed to cite it, this is a nice paper by Javier Igea from last year, if I recall correctly, showing that rates of diversification accelerate towards the poles. And this kind of all fits the same picture. The angiosperms originated in tropical environments, were kind of laying around there for a while, and after 40, 60 million years, they were able to invade and diversify into this non-tropical environment. And just to give some context to this numbers, 40, 60 million years, it's around a third of the whole of angiosperm evolutionary history. So this is kind of a lot in terms of the timing of these transitions. So these results are interesting, but of course the rise of angiosperms and this difference between the Cretaceous rise of the Cretaceous origin of families and the Cenozoic diversification of extant species is interesting by its own right, but it's also very interesting because angiosperms are known to have had many consequences in the diversification dynamics of other groups of organisms, plants and animals. You can obviously think about insect pollination or animal dispersal, kind of going along in a coevolutionary process. But today I'm going to focus, I'm going to show you a couple of slides showing the effects on two major groups of plants. This is a recent paper by Fabienne Condamine and colleagues from this year. I think it's no more than 2 weeks old, in which they analyze the phylogeny of gymnosperms. They analyze the diversification dynamics of gymnosperms through time. And this is what they get. Here you have a plot showing the speciation and extinction rates of gymnosperms through time. In red, you get extinction rates, and in blue, you get speciation rates. And what's interesting is that extinction rates in gymnosperms appear to increase towards the middle and late Cretaceous, and then going into the Paleogene, there's a dramatic rise in the extinction rates in gymnosperms. And as you might remember the few slides, this has to do something with angiosperms, but it also has to do a lot with global cooling. Temperatures have been steadily dropping towards the Jurassic and Cretaceous, but then in the Paleogene, this really plummeted into really cool climates. So this is partly explained by this trend of global cooling, but it's also explained by the dramatic rise of angiosperms. Not only the rise of angiosperms in the Cretaceous, but more significantly, the rise of modern-day angiosperms during the Paleogene, which basically drove gymnosperms to extinction in this period. So basically, the rise of angiosperms in this case coincides with the decline in the diversification of gymnosperms, but under conditions of global cooling. And this, to me, tells us that all these trends of diversification and rise in crown ages and sediment ages is really basically happening under a global context, environmental context, and that probably these changes in global conditions triggered the diversification dynamics of angiosperms after the Cretaceous. Another example is one of my favorite groups of plants, tree ferns. Here is the second largest group order of ferns, of living ferns. Here you see a phylogeny of tree ferns, dated phylogeny of tree ferns, where the branches are colored by according to estimated rates of diversification. And you can see it's interesting that diversification in tree ferns, although they are really quite an old group dating back to the Triassic, probably, the diversification of extant species really took off in the Cenozoic. Again, it's a Cenozoic phenomenon. If you look at how net diversification rates have trended through time, you see in the y-axis net diversification rate, that is basically speciation minus extinction through time here in the x-axis. And the first thing you notice is a decline in tree fern diversification, again coinciding with the rise of angiosperms in the middle to late Cretaceous. This, you can go to the paleobotanical literature. Of course, this is not the correct date. It's supposed to be 1998, I think, but it doesn't matter. But if you look at how ferns behave in the fossil record, you see a steady decline of fern abundance towards the late Cretaceous. So again, this kind of fits the hypothesis of angiosperms kind of outcompeting other groups of terrestrial plants such as gymnosperms or ferns. But things are more interesting because there appears that tree ferns have had a rebound of sorts during the Paleogene. And this, you see, I think it's my hypothesis, is that this has been driven by the rise to dominance of extant angiosperms, because it was not until the end of the Cretaceous period that really angiosperms rose to dominance and started to, and all the angiosperm-dominated biomes as we know today started to develop. The first evidence of tropical rainforests as we know them today come from the Paleogene around 56 million years ago. So my hypothesis here is that the rise of angiosperms is associated with a decline, but then a rebound in the diversification of tree ferns. And this is important because it paints kind of a picture where angiosperms not only, I think I'm repeating myself, but not only outcompeted these groups of species, but then kind of set up the stage for their diversification. Most of these species are species that thrive in angiosperm-dominated cloud forests, mountain forests, tropical rainforests. So it kind of makes sense that the development of these environments triggered their diversification. So to wrap things up, the rise of modern-day angiosperms was geographically heterogeneous and delayed for quite a while, around 40 to 50 million years. Most phylogenetic diversity and probably many morphological and functional innovations were already in place by the Middle Cretaceous. So we are not really sure why the delay. My take is that it has something to do with the environmental context of this diversification. And finally, angiosperms now revolutionize terrestrial ecosystems, not only by exploiting several co-evolutionary pathways, such as insect pollination or animal dispersal, or by outcompeting other groups, such as gymnosperms, but they also set up the stage for the diversification of other organisms. If you think of plants, angiosperm diversification set up the diversification of ferns, of liverworts, of bryophytes. So I think at the end we have so much more questions that we need to address to understand how angiosperms have shaped our world. And with that, I would like to thank a lot of people that help this project come to term, and obviously my two amazing colleagues, Susana and Hervé, for their help, and to you for listening. And I'll take questions if there's any time left. Thank you.
Thank you so much, Santiago. This was fascinating. So we'll be taking questions in a second, but first, perhaps we can stop the recording if you